G2 is the final step of interphase before mitosis officially begins. Both of the centrosomes, each containing a pair of centrioles, have formed, and the nucleolus is clearly visible. The DNA has replicated inside the nucleus and is ready to be condensed into individual chromosomes. The first step of mitosis, prophase, has begun. Here you can see that not only has the nucleolus vanished, but the DNA has been tightly wound to form chromosomes composed of two sister chromatids. The two centrosomes start heading in opposite directions as the mitotic spindles that extend from them lengthen. Prometaphase is the next stage. The nuclear envelope has disintegrated into small pieces, allowing the now even more condensed chromosomes to float freely. The microtubules have grown much longer, and some reach forward to attach to the kinetochores of the chromosomes. After prometaphase, metaphase begins. It is the longest stage of the whole process of mitosis. All of the chromosomes are arranged neatly on an imaginary line known as the metaphase plate. Microtubules extend from the centrosomes, which have positioned themselves at opposite poles of the cell and connect to the sister chromatids' kinetochores. When metaphase finally ends, the quick stage of anaphase follows. The cohesins that glued the sister chromatids together are cleaved, permitting each chromatid to separate and become individual chromosomes. The microtubules attached to the kinetochores, or the kinetochore microtubules, start to shorten, each dragging the chromosomes to opposite sides. Both ends of the cell will have the same number of chromosomes. The non-kinetochore microtubules continue to grow, elongating the cell. Telophase and cytokinesis end mitosis. During telophase, two nuclei and nucleoli take shape in each end of the now elongated cell, and the chromosomes loosen into less and less condensed forms. Mitosis has officially ended, and cytokinesis, the division of the cell, soon follows.
Two diploid daughter cells appear shortly after the end of mitosis. Each cell has 6 chromosomes, making them genetically identical to the parent cell. Meiosis 1, homologous chromosomes separate. In prophase 1, the chromosomes begin to condense. Homologs go through synapsis, which causes the crossing over. When synapsis ends, the chromosomes begin to move apart slightly. There is movement of the centrosome, the spindle forms, and the nuclear envelope breaks down, just as it did in mitosis. Later in the stage, microtubules from one pole or the other attach to the two kinetochores. The pairs of chromosomes are arranged on the metaphase plate, with one chromosome in each pair facing each pole. Both chromatids of one homolog are attached to the kinetochore microtubules from one pole.
Breaking apart inside, breaking apart inside.
Homologs are allowed to split due to the breakdown of proteins responsible for sister chromatid cohesion. The homologs then move toward opposite poles, guided by the spindle apparatus. Sister chromatid cohesion persists at the centromere, causing chromatids to move as a whole toward the same pole.
Maybe we're better off this way. It's better that we break.
By now, each half of the cell has a complete haploid set of replicated chromosomes. Cytokinesis forms 2 haploid daughter cells. Meiosis 2, sister chromatids separate. Prophase 2, a spindle apparatus forms and the chromosomes move toward the metaphase 2 plate.
Nobody gonna love me better. I must stick with you forever. Nobody gonna take me higher. I must stick with you.
In metaphase 2, similar to mitosis, the chromosomes are positioned on the imaginary metaphase plate. Due to the crossing over in meiosis 1, the sister chromatids are not genetically identical, and that is why they look different. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. Breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids then move toward opposite poles as individual chromosomes.
I wanna see you out that door, baby, bye, bye, bye.
Nuclei form and the chromosomes begin descending. Meiosis produces 4 haploid daughter cells, each with a set of 3 unreplicated chromosomes. Each of the 4 daughter cells is genetically distinct from the other daughter cells and from the parent cell.
This is the end, beautiful friend.